In this video, we'll focus on how microarrays, or also known as DNA chips, can quantify global mRNA expression. We'll spend time talking about two formats of microarrays, spotted microarrays and Affymetrix chips. We'll go over the 5 steps of a microarray experiment, and we'll talk a little bit about labeling reactions to prepare the mRNA for analysis. So using either format, the goal is to compare two samples. For instance, an example may be a control cell sample and a diseased sample. But we'll do it in different ways. In the case of spotted microarrays, we'll label one sample with one color, label the other sample with another color, mix them together, and analyze them on a single chip. For the Affymetrix type experiments, we'll keep the two samples separate, we'll label them the same way, and we'll put them onto different chips and record the fluorescence levels independently. In both cases, then we can calculate a ratio of the RNA level expression between two samples. So here are the stages for a spotted microarray. The analysis of our individual samples will be step 3. We have a test sample and a reference sample. They'll be labeled red and green. They'll be mixed and they'll be applied to a chip, which is going to hybridize with probe sequences that are on the surface, and then we will eventually observe the fluorescence using laser excitation at each of the spots, and then we can superimpose the two different colors to get a composite image, and we'll be able to evaluate the relative expression levels. Before we get to step 3 with spotted microarrays, we have to make them. And the first step, which can take quite a bit of time, is to do a PCR amplification of each gene that you want to test. So in an organism like humans, you would have to do 22,000 PCR reactions using 22,000 sets of PCR primers. So this is an expensive proposition. Then with a robot, you're going to put a little bit of the PCR product down at each of these spots so that it will be available for hybridizing with your test samples. And here we just see a description of the items on the previous page, which I won't go into any further detail right now. So again, this is an overview of the experiment. We have two different sample types, in this case normal tissue and a diseased tissue. We'll isolate the mRNA and we'll label them with fluorescent molecules, green for the control sample, red for the disease sample. Now we have our spotted microarray, and each spot is a different gene. This is gene 1, gene 2, gene 3, gene 4. Those are different PCR products. Now the mRNA that represent gene 1 will hybridize at this spot, and here you can see it's a red color, so that indicates that there is more of that gene in this sample, in the disease sample, than in the control sample. For gene 3, we see that it has a green color, and that means there's more of that mRNA in the control sample than in the disease sample. And then for any of these genes that have yellow color, that means they're present in approximately the same amount. The thing to remember is this is sort of like doing 22,000 northerns at a single time, because the probe sequences here are going to interact only with the complementary sequences in the samples, and it won't have, to first order, any effect on the interaction between this probe sequence, the third probe sequence with the RNAs in there. So you can simultaneously, in parallel, essentially do northerns for every gene in the genome. And this slide is showing here at the end a final, again, DNA chip of the spotted microarrays, and this is just making the point that for this particular spot we have a PCR product that is this sequence, which corresponds to a particular gene in the organism being studied. Now, this PCR product in this spot is present in millions of copies, but it's the only place on the chip where this gene is being queried. The spots next to it are completely different sequences that originate from different genes. On this slide, we see the labeling reaction for spotted microarrays, which is fairly simple. We have purified mRNA, which we obtain by poly-A selection. We'll use the enzyme reverse transcriptase to make a copy of cDNA, but in addition to the normal nucleotides, we'll also have a little bit of the amino allyl nucleotides, which is a small molecule here, which later on can be chemically coupled to fluorescent dyes. The kind of dyes that are used in microarrays are Cy5 or Cy3, and generically referred to as Cy dyes. The strategy behind this approach is that this amino allyl group is relatively small. It's not that disruptive to the polymerase, or incorporating lots of them into a cDNA molecule. But then after the enzymatic step is done, you can chemically couple these very large fluorescent molecules and have a labeled cDNA molecule. So this is just making it clear pictorially what happens at the surface of the chip. We have nucleic acids that are attached to the surface. Then we have, these are the probes. Then we have our labeled reference or control sample. It's complementary to the probe sequences it'll bind, allowing a fluorescent signal to concentrate at a particular location. And again, on each one of these spots, and we know the identity of the gene at each spot from our construction of the chip, these hybridization reactions are going on simultaneously for both the red and the green labeled mRNA molecules. Now this slide is just belaboring the point about the colors of the spots. We've talked pretty much about the green and the red spots when the gene is at higher levels in one of the two samples. We'd also mentioned yellow when they're at the same level, but they can also be black where there's not much of it in either sample, but it's still present at the same amounts, just at very low levels. So the yellow and black is sort of a continuum of the situation where neither green nor red predominates. If you want to see an excellent animation about doing microarray experiments, I encourage you to go to this link, which is also linked on our class homepage. So now we'll talk a little bit about Affymetrix chips. Affymetrix chips are made very different from spotted microarrays. Again, in spotted microarrays, we were doing PCR reactions in tubes and then physically depositing a little bit of that DNA onto the surface. In this case, we're going to use photolithography to enable light-directed synthesis of the DNA probes right on the chip. On the spotted microarray, our PCR products were hundreds, maybe 1,000 base pairs long. On these Affymetrix chips, we're going to have short oligos, maybe 20-25 base pairs, and they're going to be built up one nucleotide at a time directly on the chip. The light will help direct when a nucleotide is to be added so that you can do in parallel synthesis of thousands of spots based using methods developed for the semiconductor industry. So this is a slide which makes the photolithographic synthesis steps a little more clear. We have a 1 centimeter square chip, and we have actually millions of spots, but on each of these spots that there's a blocking compound which is going to prevent any nucleotides from being added. Then we'll create a mask, shine light on the sample, because in the areas where the mask is not covering the chip, we're going to remove the protecting groups, and now we will flow in the nucleotide adenine, and that will be chemically coupled at those locations. Then in the second step, we'll put down a new mask, we'll deprotect certain areas, we will then flow in thymine base, and it will be chemically coupled there. So then after several steps, we're building up nucleotides on each of the spots, and although it's used a very simple mask here, since it's light addressable, you can have it with very small features, each being independent, and we're going to develop millions of different 20 to 25 base pair sequences on a chip. And each one of these spots will have, again, millions of copies of the same strand. So it's each spot that is different. Each spot itself has many copies of a single sequence. And I was using spot, but I guess the more appropriate term is called feature. These features at this point were 5 microns on a side. Now I think they're down to about 2 microns a side. And there are millions of probes in this 25 micron, square micron feature. Now in spotted microarrays, you sort of had built-in quality control because whatever the abnormalities of printing that PCR product, both samples are going to experience the same deficiencies and be affected in a similar way. So that's sort of allowing for imperfections to still give you reliable data. With Affymetrix chips, we're using different chips for different samples, and so quality control becomes extremely important if you're going to be able to compare two chips. And the way Affymetrix has approached this problem is for every feature that they put down, there's going to be a control spot which is very closely related but a little different, with the idea being that anything that's going to affect this spot will also affect this spot a little bit. So let's talk about what each of these spots are. Remember, genes can be thousands of base pairs long. These spots themselves are 20 to 25 base pairs, so in order to cover a large section of the gene, we're going to use many features per gene. In this case, there are about 20 features for the gene, and it's spread out across the gene with a slight predisposition towards the 3' end, because those will be better represented when you're doing reverse transcription to label the mRNA. So we have about 20 features for one gene and 20 control features. The control feature is the exact same sequence as the sequence for the gene, except with one mismatch here in the center. Now the idea about putting a mismatch in this feature by incorporating a C instead of a G on the surface is to prevent specific hybridization with the gene sequence. That will only occur at the what's called the perfect match site. At the mismatch site, things will stick randomly and non-specifically, and the idea is that if RNAs are sticking non-specifically, they're just as likely to stick to either of these two sequences or locations. Furthermore, the mismatch is chosen to be right in the center of the oligo because that is the single most destabilizing spot in any hybridization event. And here's a blow-up of a perfect match and a mismatch feature. You can see the higher intensity for the perfect match. This is indicating that there's a high level of gene expression. There are different pixels here that the fluorescence imager has gotten from this feature, and a value will be assigned to this feature based on a certain algorithm on this feature. Highs and lows will be thrown out, and it'll probably use the median here. So, and then finally, the specific amount of intensity for this feature on the gene will be all the fluorescence at the perfect match site subtracted by the non-specific binding at the mismatch site. Now the labeling of a sample for Affymetrix chips is a little more involved than that for spotted microarrays. Spotted microarrays was pretty simple. We took the mRNA, we did a reverse transcription which incorporated amino allyl nucleotides, and then we chemically made them fluorescent. In Affymetrix chips, we're going to take total RNA, we'll take oligo DT primers, we will then do reverse transcription and second strand synthesis, so we'll have double-stranded DNA. On the primer that we used for the reverse transcription, we're also going to incorporate a promoter so that we can transcribe the sequence. And so we're going to make a copy of RNA from our double-stranded cDNA, and as we do that, instead of amino allyl nucleotides, we're going to use biotinylated nucleotides. And so we're going to incorporate a number of biotins into the RNA molecule. This is an amplification step, and it also provides a simple mechanism for fragmentation. You can fragment this RNA molecule in the presence of magnesium simply by heating it for a controlled amount of time. Then the hybridization will be done to the Affymetrix chip with just this biotinylated cRNA. Once it's bound, then you will flow in a molecule called streptavidin, which binds to biotin very tightly, and in this case there's a lot of fluorescent molecules that are conjugated to the streptavidin, and we'll be able to detect fluorescence then at this feature site based on the number of chromophores that are present. And here's a detailed image of one gene's expression pattern as analyzed by about 15 different features. As you can see, in each probe pairs being vertical here, that the perfect match hybridizes with a greater intensity than the mismatch. And there are a variety of formulas then that can use this fluorescence intensity to come up with a single number that is supposed to be representative of gene expression. So then again, summarizing the Affymetrix experiments, each experiment is a single color experiment, so we're going to need 2 chips. Some of the advantages is that they're commercially available, produced in very high quality, and it'll be the same type of chips that everybody else across the world is using to analyze the samples, making results more comparable. The chips are pretty expensive, but not really when you consider all the labor involved to make the spotted microarrays, but you are limited to the organisms that Affymetrix, which I should have mentioned is a company that makes these. So the Affymetrix company makes chips for a large number of organisms, but perhaps not all. If you study an organism which they don't provide chips for, then you will have to make your own spotted microarray. Okay, that summarizes the 2 different microarray formats. Now we'll talk about data analysis in the next video. Thank you for listening.